Okay, so I'd like to thank the members of the conference committee and the chair of the symposium for the opportunity. What my lab studies is innate survival behaviors, such as ingestion, thermoregulation, and aggression. How the brain is able to sense specific stimuli that triggers or moderates these behaviors from both within and outside the body, and how the brain integrates and transforms the multiple streams of information to appropriate behavior outputs, such as eating, turning on a heater, or attack. For today's talk, I'm gonna tell you about our work on thermoregulation, specifically thermoregulatory behavior, as this mouse is doing here now. This mouse is turning on a heat lamp. It is doing it by poking its nose into the hole. So in this, what we call as a self-feeding operant task, each nosepoke response will turn on the heat lamp for 2 seconds. And the entire chamber is inside a refrigerator, so it's very cold. And the mouse is highly motivated to voluntarily perform this operant task, okay? So thermoregulatory behavior is one of the most basic motivated behavior for body temperature homeostasis. And this behavior is conserved across the animal kingdom. And of course, this is because maintaining body temperature within a certain range is very critical for survival. For most animals, this range is usually very narrow, around 37 degrees Celsius or 100 degrees Fahrenheit. And significant deviations of body core temperature from this range results in impairments in molecular and cellular functions and could also cost life in extreme cases. In face of a thermal challenge, the brain prompts an array of autonomic and behavioral responses to maintain thermal homeostasis. And here I'm showing you 3 examples of autonomic responses to cold exposure. Shivering thermogenesis in the skeletal muscle, non-shivering thermogenesis in brown adipose tissues, and cutaneous vasoconstriction that reduces heat loss. These are highly stereotyped involuntary responses. The neural circuit mechanism underlying autonomic thermoregulation has been pretty extensively studied over the past few decades. The many discoveries led by the pioneering researchers, in particular Sean Morrison and Kazuhiro Nakamura's works, are remarkable. And the field has been recently rapidly expanding in the recent years by the contribution of these seasoned as well as young neuroscientists. So the scheme here summarizes our current understanding of the autonomic thermoregulatory responses. The ambient temperature detected from the skin is transmitted to the brain via the spinal cord. And the thermosensory information first reaches the parabrachial nucleus, which relays this information to the preoptic area. The POA, preoptic area, then connects to several hypothalamic subnuclei, then to the hindbrain visceral motor neurons, and then to the sympathetic preganglion neurons, which then triggers the thermogenic responses at the end organs, the muscle, the brain adipose tissue, and the blood vessels. In contrast to the autonomic thermoregulatory responses, which are stereotyped and relatively well studied, as we have just seen, thermoregulatory behavior is a motivated, goal-directed behavior that is flexibly manifested in diverse forms, such as moving to a warm place, or building nests, or performing operant responses to turn on the heater, drinking hot water, or wearing jackets in humans too. So the behavioral strategies to cope with thermal challenges are more energy efficient, cost less ATP molecules than autonomic processes, and thus represent a preferred mode of thermoregulation in animals. But despite its fundamental importance, its underlying neural mechanisms remain strikingly unclear. Even the involved brain regions, I mean, I'm not talking about the cell types, the brain regions are in macro scale are unclear. So the situation is in stark contrast with the extensively studied autonomic thermoregulation, and specifically no forebrain region required for thermoregulatory behavior, not to mention the identity of the underlying specific neurons has been identified yet. So this is very surprising if we think about the remarkable progress in our understanding of the neural mechanisms of diverse motivated behaviors over the past few decades. So we sought to first identify a brain region, a forebrain region that is required and that plays critical roles in thermoregulatory behaviors. And for this, we focused on the lateral hypothalamus, LH, which is a pivotal structure for diverse motivated behaviors. Because thermoregulatory behavior is also a motivated behavior, I said it's one of the most basic fundamental motivated behavior. We thought it is very likely that the LH would also play an important role in thermoregulatory behavior. In fact, there's a lesion study from 1970s which showed that LH lesions impaired operant thermoregulatory behavior. But this result was soon refuted by other studies showing that LH lesions have no effect or even it enhances thermoregulatory behavior. So due to this controversy, the LH has not been considered as a region that is required for behavioral thermoregulation. But the LH is a highly heterogeneous structure, both anatomically and functionally. It is made up of many tens of types of neurons that have been implicated in a number of different functions. And there are many attempts to illuminate functional heterogeneity of the LH, including the single-cell RNA-seq study. Classic lesion experiments would have indiscriminately affected some of these neurons in the LH, and perhaps that's why they observed contradictory results. So we decided to go more cell type specific, and among the many cell types in the LH, we hypothesized that the GABAergic Vgat-expressing neurons in the LH may represent a critical neural substrate required for behavioral thermoregulation. And this was because LHVgmt neurons have been implicated in these diverse motivated behaviors, including feeding. So to test this hypothesis, we first chemogenetically inhibited LHVgmt neurons during a variety of thermoregulatory behaviors. First, we found that the self-feeding operant task was significantly impaired upon the inhibition of LHVgmt neurons. So intact activity of the LHVgmt neurons are required for this operant type of thermoregulatory behavior. We also found that inhibiting LHVgmt neurons impaired the selection of the optimal temperature in a linear chamber with a floor thermal gradient. And this is an instinctive type of thermoregulatory behavior that does not require training or learning. Control mice primarily occupied the position corresponding to the thermal neutrality, the optimal temperature, but the experimental group of mice failed to do so. And we also examined nest building, which is another instinctive cold defense behavior in mice. And here, control mice robustly shredded pressed cotton squares to build a nest at a cold ambient temperature. But the experimental group of mice did not exhibit this nest building behavior. We further examined the postural extension, which is a thermoregulatory behavior observed at high ambient temperatures. And as you can see here, inhibiting LHVgmt neurons also impaired postural extension. So chemogenetic inhibition of LHVgmt neurons impairs diverse thermoregulatory behaviors. And I'm not going to show you the data, but these effects cannot be attributed to motor deficit. And inhibiting LHVgmt neurons did not affect autonomic thermoregulatory responses. So together, our results identify LHVgmt neurons as a neural substrate necessary for behavioral thermoregulation, but not for autonomic thermoregulation. And I can also go on and say that to the best of our knowledge, this is the first identification of a forebrain required for diverse thermoregulatory behaviors, forebrain regions, and also the forebrain cell type. So if these neurons are required for behavioral thermoregulation, these neurons may encode the key aspects of thermoregulatory behavior, such as motivating properties of the thermal stimulus and maybe the behavioral response to thermoregulatory behavior itself. So we next examine the in vivo real-time activity of the LHVgf population using vibrophotometry. And during the self-feeding operant task, we observed a very interesting pattern of activity. The LHVgf population was activated by the notebook, but then this population activity was potently inhibited by the subsequently delivered heat stimulus. And because the heat stimulus provided at cold ambient temperature is a reward, we we conclude that the LH-Vgat population is overall excited during the operant thermoregulatory behavior, but is then inhibited by thermal reward. The inhibitory responses of LH-Vgat neurons to thermal reward was unexpected, given the previous studies showing that the consumption of caloric reward activates LH-Vgat neurons. And in line with the previous findings, we also, in our hand, found that this population is activated by the consumption of caloric reward. So in summary, these results show that the overall activity of LH-Vgat neurons is increased during thermoregulatory behavior, and is also increased during the consumption of caloric reward, but is suppressed by the consumption of thermal reward, surprisingly. So LH-Vgat neurons are not only required for behavioral thermoregulation, but also encodes key aspects of thermoregulatory behavior. Okay, so to extend the finding that the population activity of LH-Vgat neurons is inhibited by thermal reward, we systematically examined the responses of the heat stimulus, responses to the heat stimulus at 4 different ambient temperatures, from cold to hot or from low to high. And while the heat delivery is rewarding at low ambient temperatures, the heat stimulus becomes aversive at high ambient temperatures. And as you can see, this population was strongly inhibited by the heat stimulus at 8 Celsius. Again, it was inhibited by thermal reward. But strikingly, the responses to the heat stimulus was reversed to excitation at high ambient temperatures. So apparently, the overall activity of LH-Vgat neurons is suppressed by thermal reward and is activated by thermal punishment. And we further tested if the similar responses would be elicited with a cold stimulus, and it did. The LH-Vgat population was potently activated upon the cold air delivery at the ambient temperature of 8 Celsius. And in contrast, the population activity was suppressed by the same cold air delivery at the ambient temperature of 37 Celsius. So together, our data show that the LH-Vgat population bidirectionally encodes thermal punishment and reward. And I'm going to call it simply PNR, punishment and reward, for convenience. And this result holds regardless of the temperature of the thermal stimulus itself. Since motivated behaviors arise in 2 forms, seeking rewards and avoiding punishments, the bidirectional encoding of thermal punishment and reward by LH-Vgat neurons may critically support behavioral thermoregulation. But these interpretations are all derived from the fiber photometry recordings, the overall activity of the LH-Vgat population. And we don't know how these distinct stimuli and behavior are encoded at the single cell level. So we turn to two-photon microscopy. We image the calcium activity of individual LH-Vgat neurons. And I'm not going to be able to show you all of our data and analyses today, but just to highlight you the most important findings. We first found that about a third of LH-Vgat neurons bidirectionally encode thermal PNR, the thermal punishment and reward. And roughly another third of LH-Vgat neurons encode caloric reward. And the rest of them, which correspond to another roughly third of LH-Vgat neurons, were not responsive. So this purple area showed the overlap between the neurons excited by thermal punishment and those inhibited by thermal reward, which is the definition of the thermal PNR encoding subpopulation here. And importantly, this Venn diagram shows that the thermal PNR encoding subpopulation was largely separate from caloric reward encoding LH-Vgat subpopulation. So these data demonstrate the presence of at least 2 functionally distinct LH-Vgat subpopulations, namely the thermal PNR subpopulation, the purple one, and the caloric reward encoding subpopulation, the green one, which were lumped together in the fiber photometry recordings, but now dissociated with the help of the single cell 2-photon calcium imaging. And one thing to add is that the thermal PNR neurons were not unspecifically activated by any kinds of punishment, because these neurons were largely silent to the quinine delivery in the mouth. And most thermal PNR neurons were also not inhibited by turning off the electric push-shock. So these results suggest at least some degree of specificity to thermal aversive and rewarding thermal stimulus, not to any kinds of rewarding and aversive stimuli. And importantly, we found that a significant proportion of the thermal PNR encoding neurons were recruited during thermoregulatory behavior. So in this experiment, mice were trained in a head-fixed version of the self-eating operant task, in which a lever press triggered the heat reward delivery. And we observed that the thermal PNR neurons were overlapped with the lever press activated neurons significantly more than by chance. So these results indicate that the thermal PNR encoding LHVgf subpopulation is engaged during thermoregulatory behavior, suggesting that the thermal PNR neurons likely play a critical role in guiding thermoregulatory behavior. So we then asked how LHVgf neurons would receive thermosensory information that must be needed to appropriately guide thermoregulatory behavior, and also for encoding the valence of thermosensory information. So we mapped the whole brain inputs of LHVgf neurons and found that these neurons receive monosynaptic inputs from basically all over the brain. But towards the hindbrain, what's notable was that they receive strong direct inputs from the parabrachial nucleus, especially the lateral part of the parabrachial nucleus, LPB. And the LPB is the region that is very well established to relay thermosensory information from the body to the brain. It contains both warm-sensitive and cold-sensitive neurons, and lesions in the LPB impairs the both behavioral and autonomic modes of thermoregulation. We also have the relevant data, but I'm going to skip on that for the sake of time. But anyhow, for these reasons, we consider the possibility that the thermosensory inputs from the LPB to the LH would also play a key role in thermoregulatory behavior. And again, to make a long story short, we found that chemogenetically inhibiting the LPB to LH projection selectively suppressed operant thermoregulatory behavior, but not an equivalent of operant task for the caloric reward. Additionally, this manipulation did not affect autonomic thermoregulatory responses. So the LPB to LH pathway represents another neural substrate that is required for behavioral thermoregulation. And furthermore, we also found that inhibiting the LPB soma abolished the neural encoding of thermal reward by LHVgf neurons. And this was selective to the encoding of thermal reward because the encoding of caloric reward was not affected. So together, our results demonstrate the thermosensory parabrachial inputs are required for both thermoregulatory behavior and also for the neural encoding of thermal reward by LHVgf neurons. Okay, so to summarize today's talk, we first identified LHVgf neurons as a neural substrate required for diverse thermoregulatory behaviors. And via fiber photometry recordings and two-photon calcium imaging, we revealed a subpopulation of LHVgf neurons that bidirectionally encode thermal PNR, punishment and reward, and that are engaged during thermoregulatory behavior. And this thermal PNR subpopulation was largely separate from caloric reward encoding LHVgf subpopulation. And we showed that thermosensory input from the lateral parabrachial nucleus is required for both thermoregulatory behavior and also the encoding of thermal reward by LHVgf neurons. So together, our data establish LHVgf neurons as a critical neural substrate for behavioral thermoregulation and point to a specific role for the thermal punishment and reward encoding LHVgf subpopulation and thermoregulatory behavior. Okay, so with that, I would like to close my talk. I thank the KLab temp team leaders, Xiun, Myungsun, and Dongyun for their wonderful job, and also for other team members. And I also acknowledge my collaborators and funding sources. And thank you so much for paying attention to the screen for a long time. Thank you.